Hello, okay, Sec 3 Bio students. Okay, so today we're going to go through the Nutrition in Plants worksheet. So if you take a look at the question number 1, you are expected to recognize and identify the different cells that are present in the leaf and to know the different adaptations. Now do note that the labeling line ends on a single cell. So you should be naming D as upper epidermal cell instead of upper epidermis. Okay, so similarly E, F, if you say that it's palisade mesophyll layer, it will not be accepted. Now do note that the gut cell should have some chloroplasts that can be seen, okay, but in this diagram we can't see them. Now I will bring you to part C to just highlight the answer to you. That it's important to write that the gut cells regulates the rate of the inward and outward movement of the following gases instead of regulating the inward and outward movement of the gases. That's all. Because if you the stomata does functions very differently as it doesn't function like a partially permeable membrane that can regulate the inward outward movement based on size, okay, but they control the rate, how fast, and it's based on concentration gradient. So in this leaf, if it's photosynthesizing and they happen to produce like 50% of oxygen over here, so an atmospheric level of oxygen is about 20%. So overall there is higher concentration of oxygen gas in the intercellular air spaces and it will diffuse out. Okay, so moving on to question 2. Now in question 2 you can see this leaf that you should, as proportionately as possible, okay, identify the areas that will turn iodine solution blue-black and the brown. Okay, so what happens is that your labeling line should be drawn using ruler and pencil, and your you should not shade the diagram at all. So you can see I'm not shading, I'm just using I'm using freehand to draw, but you should use a ruler. Okay, and the label should be written in ink. So these are the regions that will turn blue, black, and the other region is where iodine solution will remain brown. Okay, so now if you take a look at question number 3. Now in question number 3, you are given a aquatic plant that is provided with light. So this aquatic plant is able to photosynthesize and it produces oxygen gas and is bubbled up its way, and the gases are being collected here. Now if you take note of the trend, as I increase temperature from 15 degrees Celsius to 40, the number of bubbles per minute actually increases. But when I increase the temperature from 45 to 50 degrees Celsius, the rate of bubbling actually decreases. Now, so this reminds you of enzymes, I hope, okay? Because photosynthesis is an enzyme-controlled reaction, and we have learned how enzymes work, and I hope that you can see the trend follows the trend of the rate of enzyme activity with temperature. So you are supposed to draw the graph. Okay, now I think when you try this will be the best scale that you can use, but you will not be able to occupy 2/3 the space of this given graph, but you can at least occupy half the space. And do remember, when you draw best fit graph, it should both points, okay, that can't fall on the line, it should be balanced out. Okay, so a rough idea of it will be like that. Now can you must you always start the both x's on 0? The answer is no. And this is, you can see that I started my x-axis, okay, with 15. If you start with any other figure, your graph might be too small. So take note of this. Your teachers will actually take a look at your physical answers as well for this graph. Now you can see that the answers, okay, based on the questions is number 1, describe and explain. So do note the question command over there. You have to indicate, take note, okay, of the units, okay, and you need to explain it based on your knowledge of how enzyme works. So you see similar keywords that appear for enzymatic reaction from your previous topic on enzymes. So moving on, okay, you can see the rest of the questions with their answers. Feel free to discuss any of your answers with your teachers, but yeah, I think the answers are really quite self-explanatory. Now for the last question, second last question. Now this student was carrying out an experiment. So what the student did was to punch 36 discs from a leaf and place them in water this way, and it's connected to a suction pump. Now how does the suction pump work? It's connected to a machine that will suck the air that is present in the conical flask out, and it will also force whatever air molecules that are gaseous molecules that are present in the intercellular air spaces of the leaf disc out of the leaf. So when all these gaseous molecules are removed from the leaf becomes less dense and they will start to sink. Because the air is removed, the air will actually make them less dense. So what happens, they are placed into sodium hydrogen carbonate solution, which gives them carbon dioxide gas. Okay, so the leaf is given sunlight and photosynthesis will occur. So as photosynthesis occur, oxygen gas is produced and is accumulated again in the intercellular air spaces, which results in the discs being less dense, and they will start to float to the surface again. So this student actually time, okay, the time that is required for the disc to float to the surface. Now you can see that the timing is converted to rate by taking 1000 divided by the time that is taken. Okay, the unit, since I take, okay, so time was recorded in the seconds, so the rate, I took 1000 divided by second. So you learn in math that when the seconds are in the denominator, when I bring it up, it will just be s minus 1, and that is why the units is s superscript minus 1. Okay, now why 1000? Why this golden number? Okay, it is normally explained, given in the experiment. Sometimes it is 1 divided by time, sometimes 1000 divided by time. It's based on it's based on the values that that is obtained and how to get good values to actually plot easily. Okay, so for this, do not worry about when to use what golden number to divide. Usually it's stated in the question. Okay, do take note that this part A asks for the word equation. If you do not write the conditions required, you will be penalized. Okay, there's no half mark given at all times. Okay, then the explanation for part C is very important as you figure out how to explain in words why the bubbles came out from the leaf disc at first and why did the disc eventually float. So take note, just now as I said, the oxygen filled up the intercellular air spaces. Okay, the oxygen is not at the stomata because when it's at the stomata, it will not cause the leaf disc to be less dense. Okay, it's already outside the leaf. So the answer that the oxygen fill up the air spaces is very important. Okay, now and part D is about the limiting. Part D and E, no, it's only part D. Sorry, it only shows you the limiting factors. Okay, when is it limiting and when is it not? And remember that the graph overall will actually look like when the rate of photosynthesis with concentration of carbon dioxide gas. Okay, the graph looks like that, which you have been informed how to read this graph. So at the start, when the concentration of carbon dioxide gas is not very high, the rate of photosynthesis will increase when you increase the concentration of carbon dioxide gas. But when you increase when the concentration of carbon dioxide gas is very, very high already, okay, the rate of photosynthesis doesn't depend on the concentration of carbon dioxide gas, but it depends on other things, other factors like light intensity and temperature. Okay, so environmental conditions, so these will be the only acceptable answers. Now with that, let's take a look at the last question. Okay, for this last question, you have to understand the nature of bacteria. Okay, so a lot of information is given in the question which you are expected to read very carefully. So algae and bacteria, they respire aerobically. Okay, meaning that normally in the dark, they will take in oxygen, but they will give out carbon dioxide gas. Now, then what they do is they shine a white light through a prism. Now, what does the prism do is to separate the light, which consists of 7 colors, okay, out. So this is the order that you see the different light rays. Okay, so red is being shown here, okay, then orange here respectively. Okay, now you'll realize that actually the things that we see, they are the colors that we see is because they reflect that light. So green leaves appear green because they reflect green light, but they absorb all the other light rays. So bacteria, okay, informed over here, okay, they are motile, and the definition for motile is given here, means that they will move. So at the start, in the dark, okay, the algae and bacteria, they were respiring, okay, and there is no oxygen that's being produced by the algae. So the bacteria will actually be spread out evenly throughout all the cells. Okay, the bacteria is all throughout like that. Okay, instead, then once you shine the light, then what happens is that the bacteria start to move, and it shows the movement as you can see in the diagram already. So they move more towards the regions, the extreme regions that you see over there. So sorry, let me scroll back. Okay, so they move to these 2 regions mainly. Okay, So now, what happens is you are supposed to understand and explain what is happening over here. So for the first question, based on your results, identify two wavelengths of light that is most effective. So you can see wherever there is more bacteria, means that wavelength is used more, okay, for photosynthesis. So how do you conclude? Okay, it's because you show the two regions produce the largest amount of oxygen. Now, for part B, explain why it is important to place the algae and bacteria in the dark for 24 hours before the start of an experiment. It's the same reason why we place the plants in the dark, okay, to destarch in your investigations that you have learned in your textbook. So you make sure that they are not photosynthesizing at all. Okay, so whatever oxygen that is being produced is because it is from the experiment. That the bacteria was evenly distributed, and whatever that you see the result is because of the different rates of photosynthesis that are occurring under the different wavelength of light. Okay, with that, we have completed the entire worksheet. Now, feel free to clarify any questions that you have with your teachers. Do submit the hard copy of your worksheet too. Thank you.